Okay, hopefully everyone can see that. Thanks so much for inviting me to come out, through Zoom to give a seminar. I really appreciate the invite, and I hope you enjoy what I have to share with you today. So, as Cole mentioned, I'll be discussing the role of food in the snowshoe hare cycle. But first, we'll start with some background. So, what determines species abundance? This is a question that comes up a lot in ecology. And in order to identify potential controlling factors, we need to determine the differences between increasing and declining populations. And addressing this question really improves our ability to predict numerical changes into the future and is critical for conserving and protecting species. Now, much of this research has been done on cyclic species, which naturally experience major fluctuations in abundance and provide a unique opportunity to study limiting and regulating factors. Population cycles are a remarkable phenomenon in ecology and occur in numerous species around the globe. And probably one of the most famous examples, and the first to be empirically demonstrated, occurs between the snowshoe hare and the Canada lynx in the boreal forests of North America. Now, snowshoe hares are a medium-sized herbivore and are often considered a keystone species in the boreal forest. And this is due to their impact on all the other species in the forest, particularly the Canada lynx, who is considered a specialist and specializes on consuming them. Now, snowshoe hare abundance fluctuates dramatically with 10-year periodicity. For context, if you set up 60 traps in the forest when the density is high, odds are you could trap as many as 60 hares. But if you do the same thing when the density is low, you're lucky to catch even one hare. So we're really talking major fluctuations. And the cycle is characterized by demographic changes throughout different phases of the cycle. And so the cycle is separated into 4 main phases based on these demographic changes. During the increase phase, the population grows rapidly, and survival and reproduction are considered to be high. At the peak phase, the population halts its growth and shifts from population growth to population decline. And at this point, survival and reproduction have decreased. Now, in the decline phase, the population abundance drops dramatically. Survival and reproduction are also very low at this time, with overwinter survival being as low as 20% and reproduction dropping by about 50%, with hares producing 2 litters in a summer instead of their usual 4 litters, which they're capable of when things are good. By the time we get into the low phase, the population decline is halted. Now, survival has recovered at this point, but reproduction has not yet recovered and does not recover for another year or more. So the big question is, why does this happen, and what causes these major changes in survival and reproduction? Hares fluctuate with their main predator, the Canada lynx, so a first explanation would be direct predation. And it makes sense because over 95% of hares are killed by predators. However, direct predation can't account for changes in reproduction. Therefore, it is unlikely to be the sole driver of the cycle. 2 other potential drivers that have been proposed are winter food limitation and indirect effects of predation or fear effects, which is when the presence of a predator is enough to influence demography through increased stress or fear. Food may be a limiting factor for hares in terms of either quantity, quality, or accessibility, particularly during the winter months. During this time, hares rely on the small terminal twigs of shrubs, which are harder to digest and have lower nutrient content than their summer diet. In many cases, these twigs are also highly defended through plant secondary compounds. Earlier work by John Bryant showed that secondary compounds increase with plant regrowth, which means plants could be highly defended after periods of intense browsing, like the kind that occurs at the hare peak when densities are very high. Per capita changes in food availability could influence body condition, resulting in these changes in reproduction that we see in the cycle. So a reduction in high quality food could also alter behavior, causing individuals to increase foraging rates or forage in more open, risky habitat. And these changes in behavior, along with the changes in condition, could make hares more vulnerable to predation, therefore affecting survival. And so increased risky behavior could then also lead to increased encounters with predators, causing nutritionally poor individuals to have higher chronic stress. So if there is any chronic stress occurring, it could be happening in part due to food shortages and not just because of predation. Now, because of the difficulty with quantifying food on the landscape, researchers in the past have used food supplementation experiments to try to assess the impact of food on the cycle. Now, this was most famously done by Charles Krebs, Stan Booten, and Rudy Boonstra as part of the Kluane project up in the Yukon. In this experiment, they had several different treatments manipulating food and predation. And in each treatment, they measured the treatment effects on demography. So looking at their results, overall, food increased density threefold and did not affect the rate or timing of the decline in abundance. But if we look at 30-day survival rate for the food treatment, food had little effect on survival compared to control grids, which are seen in the red, and no effect on reproduction or juvenile survival. So this did not make a very strong case for food being a necessary factor in the cycle or in driving cycle dynamics. Now, since the Kluane experiment, studies by Rudy Boonstra and Michael Sheriff have linked changes in predation risk to chronic stress and have showed that this could affect reproduction. As predation risk increases through the cycle, the degree of chronic stress increases and is thought to affect reproduction. And so these studies have pushed the current consensus on what drives the cycle to a combination of direct and indirect predation, mediated through predator-induced stress. And food has been essentially dismissed as a necessary or contributing factor to the cycle. However, food may have been dismissed too quickly. There are issues with large-scale, population-wide food supplementation experiments that have been documented in 2 reviews, one done by Stan Booten and more recently by Prevedello. And these issues are important to consider when interpreting these results. First, there can be unequal access to the food, which means you could be sampling individuals for repro data that never really had additional food. There can also be increased immigration, which increases densities to a point that can cause increased competition and further unequal access to food. And you can get what's called a predator pantry effect, where the high prey density caused by additional food attracts predators and actually decreases survival. In this instance, the food supplementation area can actually become a sort of ecological trap, where the food addition site is perceived as high quality habitat, but survival and reproduction are actually reduced due to intense predator presence coupled with increased competition. This results in these studies often finding change through increased density, but no change in demography. So my main objective for this project was really to determine the degree to which food is contributing to the snowshoe hare cycle through an individual-based food supplementation experiment, with the goal of answering the following questions. Number 1, are hares food limited during any phase of the cycle? And number 2, how does food limitation influence demography? And through which mechanisms or what manner, either through behavior or condition or physiology, does food influence survival and reproduction? And I'll get into the physiology portion in the second part of the talk, but for now I'll focus really on behavior and more condition metrics. So to determine whether hares are food limited, I looked at the effects of food supplementation on survival and reproduction. And to determine the potential ways in which food acts upon hares to influence demography, I looked at the effects of food on condition and behavior through body mass, foraging and vigilance behavior, home range size, movement rates, and habitat selection. And basically, we expect that if snowshoe hares are food limited, supplemented hares will have increased survival and reproduction. And this improved demography would likely occur through increased body mass and vigilance, decreased foraging and movement, and selection of safer habitat. So in order to do this, we conducted the food supplementation experiment in the Kluane Lake area of the Yukon in Canada. It's a pretty homogeneous landscape comprised primarily of white spruce and smaller amounts of willow and birch shrubs. And the study took place across 4 winters from 2015 to 2019 and occurred in the months of November to April, except in the last year where we carried it out between January and April. Now, the density of almost all species in my study area has been monitored for over 40 years through the Community Ecological Monitoring Program that's been established by Charles Krebs and Alice Kenny. And shown here is the density of hares in this study area for the past 10 years. And highlighted in the gray box are the study years in which I worked. So I'll be referring to each of my study years by phase to make it easier, but basically I worked in the increase phase, the peak, and then the first and second years of decline. Now, during this time, predators, meaning mainly lynx and coyote, also experienced an increase in their population, followed by a decrease in 2018 and 2019. But if we look at it in terms of predator-prey ratio, you can see that it continued to increase throughout the study, from the increase phase to the second year of decline. So I use predator-prey ratio as an index of predation risk. So it's important to note that predation risk was lowest during the hare increase and highest in the second year of decline. So to collect the data, we would go out and trap hares. And we focused the study on female hares only. So we would collar and collect data from females that we captured, aiming to have a minimum of 20 collared females in each treatment. And then we would start the feeding experiment. But we had to figure out a way to supply food individually without causing the same potential biases as some of these population level experiments. So we first tried small piles and put a camera at each pile in order to monitor activity and ensure individuals were accessing food. But it turned out this wasn't good enough because even a small amount of food actually caused fights between the hares. And so we realized we needed a feeder that would block access to controlled hares. And we tried some very hilariously bad methods, from things called the Wonder Bowl to the Meow Space, before we finally settled and found the right feeder for the job. And so I use the SureFlat Pet Feeder, which is actually an indoor cat feeder, that is microchip activated and only opens if an individual has the right microchip. So this prevents controls from accessing the food and allows only food ads to get it. And so we also put cameras at the feeders to monitor activity. Additionally, by this point, we also realized that heat shrink glows in the infrared light of the cameras. So we created individual barcode patterns so that we could monitor individual feeder use and ensure that individuals, dubbed food ads were actually accessing the feeder, as well as how often. And here is a video, which I'm hoping is working through Zoom, to give you an idea of how these feeders work. Now, these feeders were set up within hare home ranges, and each hare had access to all the feeders, but most of them used between 1 and 3 feeders. So once we got the food ad up and running, we started all the monitoring and data collection to look at differences between control and supplemented hares. So to get at survival, we monitored survival daily through the winter using VHF telemetry. And we collected reproduction data through maternity pens to look at repro differences between treatments for the first litter. Now, the first litter occurs right after winter, so I predicted it to be the most strongly impacted by winter food conditions. Females tend to drop their litters anywhere and they don't make burrows or nests, so it can be almost impossible to find the babies. So each May, females close to giving birth were placed in these pens for a couple days until they gave birth. Once the babies were born, we collected data on litter size, total litter mass, as well as individual leveret mass. To look at food effects on condition of hares through winter, I looked at average late winter body mass, as well as average mass loss over winter. And to look at food effects on behavior, each female was equipped with a collar that had GPS and VHF, as well as an accelerometer. Now, accelerometers measure acceleration on 3 axes, and with proper calibration using machine learning algorithms and validations on live animals, we can distinguish differences between different behaviors. For hares, we were able to separate their behaviors into 4 main categories: not moving, hopping, sprinting, and foraging. We further separated the behaviors more recently into foraging and vigilance behavior. Now, vigilance refers to short breaks or pauses that lasted less than 5 minutes and on either side had a foraging bout. So it's basically not so much vigilance for the whole day, but of their time spent foraging, how much of that time was spent being vigilant. So with the GPS data, we were also able to look at home range size, distance traveled per day, as well as habitat selection. So using the methods I've described, to bring everyone back around, my goal again was to determine if hares are food limited by measuring survival and reproduction, and how this food may influence demography by measures of condition and behavior. So getting into the results, how did food supplementation affect survival? Well, in my study, the proximate cause of mortality in about the 275 hares that we monitored was predation, with over 99% dying due to a direct predation event. Now, when looking at the effects of food across the study, 6-month survival probability was 2 times higher in supplemented versus control hares. And survival rates in supplemented individuals were similar to those seen in the early increase phase when hare survival is its absolute highest. If we look at each year separately, based on these Kaplan-Meier survival curves, which show survival probability over time, looking at the top left first, supplemented individuals had an average survival probability 3 times higher than controls in the increase phase. And the effect was not as strong in the peak, but food significantly improved survival again in the first year of decline. But what's fascinating is you can see survival drop dramatically in both groups in the decline phase, indicating that food no longer provided a survival advantage at this time. But overall, we can say that food did significantly improve survival. So if we move into reproduction, and remember, these are results for only the first litter. If we look at litter size, or this is to be more specific, the number of offspring produced, there was no difference between control and supplemented individuals, nor was there any variation between cycle phases. So there was not much to measure here. But if we look at total litter mass, this is now the total biomass the female produced in her first litter. Supplemented hares produce significantly bigger litters in terms of total mass, by an average of about 23% across the study. Total litter mass also varied with cycle phase, with the largest mass occurring in the peak and first year of decline, but the lowest mass in the second year of the decline. And these are means with 95% confidence intervals, pretty much for every graph I'll show you kind of from here on in. Now, if we look at individual leveret mass, the mass of each individual baby, supplemented individuals produce significantly larger babies across the study, and they were on average 14% larger. So individual mass varied with cycle phase and followed a similar pattern to total litter mass, with the highest mass in the peak and the lowest in the second year of decline, particularly in control hares shown in red. So we can say that food did significantly increase reproductive output, mainly through the size of the offspring. Which answers our first question. Food did have a significant effect on hare demography, which is indicating that hares are indeed food limited. But the next question is, how did food influence demography? What was the mechanism that allowed fed hares to have improved survival and reproduction? So if we first look at body condition, snowshoe hares typically lose mass over winter. This starts in about January, and this did happen in my study as well. And if we look at individual daily mass loss or mass change shown here, which would measured as the difference in mass between at least 2 captures that were 60 days apart, you can see that on average, supplemented hares lost significantly less mass over winter, and in the peak actually gained mass, but that both groups experienced more body mass loss heading into the decline phase. So this resulted in differences in late winter body mass, which is from February until April. This is shown here with 95% confidence intervals again and means, and you can see that supplemented individuals were significantly larger by about 8% at the end of winter. Across cycle phases, we see that hares were larger in the peak, and both groups had the lowest body mass in that second year of decline. So food significantly improved condition. Now if we move into the behavioral results, first, we will look at foraging behavior. Although supplemented individuals used feeders, they still went out and foraged for natural browse. So they actually spent less than 1 hour a day at the feeders, which allowed us to still measure sort of instances of foraging outside of just feeder use. So if we look on the left at time spent foraging per day, supplemented individuals foraged approximately 1 hour less per day than controls. But what's really interesting is if you look across cycle phases, foraging time actually decreases in both groups, until in the decline phase, hares are actually foraging 2.5 hours less per day. And it's especially interesting that control hares are doing this. Now, if we look over on the right at the proportion of foraging time that hares spent vigilant, supplemented hares spent more time vigilant than controls, but only to a certain point at which behavior plateaued, which might indicate a trade-off or sort of a maximum amount of vigilance that is worth the cost of not foraging. And if you remember me mentioning that from the increase phase to the decline phase, predation risk is increasing, which suggests that hares are foraging less and spending more time vigilant when predation risk is highest, potentially indicating an interaction effect with food and predation. So we can say confidently that food significantly decreased foraging and increased vigilance. Now, if we move into movement behavior, this is sort of a distance traveled per day and home range. The plot on the left shows you home range sizes in hectares, calculated as 95% UDs in each year. And the right plot shows you distance traveled per day in meters in each year. So we predicted that food supplementation would decrease home range size and distance traveled per day. But we found food really had no influence on home range size, and there was quite a lot of significant variation across cycle phases, with home ranges in the second year of decline being more than twice the size of home ranges during the peak phase. If we look on the right panel, food supplemented individuals traveled significantly less per day by about 100 meters. And distance traveled also significantly varied among cycle phases, following the same pattern as home range size, with the least distance traveled in that peak year and greatest distance in the second year of decline. So these patterns in movement did not really track predicted changes in food or predation risk. And so we suspect that these sort of movement behaviors are more likely influenced by other factors such as snow conditions and snow depth, which matched more closely with the patterns we saw here. So the most predominant available habitat type for hares in our study area is conifer, with the next most available being shrub. And based on previous research, conifer is considered to have good cover but poor food, and shrub is considered good food but poor cover. And so to calculate habitat selection, I use selection ratios with 95% confidence intervals, looking to see if they overlap 1. And food supplemented individuals selected for conifer in the increase phase, peak phase, and in the second year of decline, whereas control individuals selected against conifer in the increase and then selected for conifer in the decline phase. If we look on the right for shrubs, the pattern is not as strong due to the large amount of variability in availability and individual differences. Food supplemented individuals used shrub habitat as it was available, and control individuals actually selected for shrub habitat in the increase phase, but not in any other phase. But this is interesting because, again, remembering that predation risk is increasing from the increase phase to the decline, it does seem like control hares are selecting for conifer as predation risk increases, which is considered better cover but worse food. Whereas supplemented individuals seem to be selecting for conifer even under low risk. So these findings suggest that hares move into safer habitat with worse food as predation risk increases. Further evidence that this could be driven by predators is that we also saw decreased feeder use by supplemented hares. So feeder use was very low in the decline, which may have been due to predators keeping hares from accessing them. This, combined with the decreased foraging and selection for conifer habitat, suggests this may be caused by predators. So even if food is available, they may not be able to access it. So in summary, food significantly improved survival and reproduction. And supplemented individuals had larger body size, lower foraging rates, higher vigilance rates, they moved less, and they selected for safer habitat. Food effects were also phase-specific, with strong effects in the increase and little effect in the decline phase. Now, food effects on survival diminished during the decline, suggesting that predation pressure may be too high and there is nothing hares can do. But food still increased reproduction during the decline phase. So if food is still playing a role in this phase, it's most likely going to be through effects on reproduction and potentially maybe through recruitment. So since food had such strong effects on behavior and condition, I next wanted to see if food supplemented individuals were also experiencing decreased levels of stress as a result of behavioral adjustments that may have led to less encounters with predators and decreased chronic stress. But before we can get into that data, we first have to understand how the stress axis works in mammals. So when an organism experiences a stressor, a signal cascade occurs through the HPA axis and results in the release of glucocorticoids, which in hares is cortisol and is often called the stress hormone. cortisol circulates in the bloodstream and passes into tissue and exerts biological effects, which is synonymous with the fight or flight response. In order to recover from an exposure to a stressor, the system has a built-in feedback loop. So once high levels of cortisol are detected in the blood, the system shuts itself down and brings it back to baseline. Having high levels of circulating cortisol for long periods of time can result in chronic stress, which can have deleterious effects on the body, like decreasing reproduction and immunity. Now, the body does have a way to buffer against cortisol, and most of the cortisol that circulates in the body is actually bound to a corticosterone binding globulin, known as CBG, that makes it inactive. So under a short-term or acute stressor, the body releases both CBG and cortisol. The CBG binds to most of the cortisol, leaving less of it free to exert biological effects on the body, and things return to baseline fairly quickly. However, under conditions of chronic stress, individuals are usually in poor condition and unable to produce as much CBG, meaning when they are presented with a stressor, the body releases a high amount of cortisol, but not much CBG. And this results in high levels of free circulating cortisol that is active in the blood and can exert deleterious effects on the body. If these levels of cortisol are high for long periods of time or frequently stimulated due to frequent stressors, such as increasing amounts of predator encounters, then this chronic stress can lead to decreased reproduction and further decrease in condition, potentially making them susceptible to predation. In snowshoe hares, the prediction is that stress levels or cortisol levels will be highest when predation risk is high, which occurs during the decline phase. This is also when earlier data suggests that hares may be foraging in a predator-sensitive manner and not able to access available resources, as we sort of covered earlier. So the predictions are that control individuals should have cortisol levels that increase from the increase phase to the decline, and CBG levels that decrease from the increase phase to the decline phase as condition worsens following changes in predation risk. But with access to high quality food, these effects may not occur due to improvements in condition and changes in behavior that led to higher survival and reproduction. So in the second part of this experiment, I wanted to determine if access to high quality food can buffer against the negative effects of chronic stress that are caused by increasing predation and potentially decreasing food availability, and to evaluate how these indicators of chronic stress changed in this cycle specifically with increasing predation risk through the increase to decline. But in order to test this, we do what's called a hormone challenge. So it allows us to measure an individual's physiological capacity to mount and terminate a response to a stressor, but without interference from the immediate stress they may be experiencing due to capture or handling. So we take a baseline or initial blood sample. We then give an injection of dexamethasone, which stimulates the feedback loop and decreases circulating cortisol. We then collect a dex bleed sample and give an injection of ACTH, which triggers the release of cortisol. And then we collect a series of serial bleeds to monitor how high the cortisol release peaks and how long the response lasts. Along with changes that occur in cortisol, chronic stress also typically increases the production and storage of glycogen, which is broken down into glucose and mobilized during a stress response, providing energy. So we expect to see high glucose levels moving from the increase to the decline phase following changes in predation risk, and lower glucose in supplemented individuals if they are not experiencing this chronic stress. So we predict to see that hares with supplemented food will have decreased free cortisol, increased CBG, and decreased glucose. And for the cycle in general, with increasing predation risk, we expect to see across the cycle increased free cortisol, decreased CBG, and increased glucose. Now, if we look at the data collected at the initial bleed, so this is the sort of first baseline sample we took, free cortisol levels did not differ between control and supplemented individuals. And across cycle phases actually showed a decreasing trend, which is the opposite of what is predicted. On the right, if we look at CBG, which, not to add confusion, but is measured as the maximum corticosterone binding capacity, or MCBC, at the initial bleed, MCBC was higher in supplemented individuals, although the effect was not significant. And across cycle phases also showed a decreasing trend, which is more consistent with the prediction made by the chronic stress hypothesis. Now, if we look at the response to hormone challenge, these graphs show the response calculated as the area under the curve in free cortisol in response to hormone challenge. And food supplementation significantly increased the response to a hormone challenge, which again is the opposite of my prediction that food would decrease the stress response. This could potentially represent a better preparedness for upcoming stressors and may indicate that if you can afford to be physiologically more prepared, maybe you should be. Now, if we look at the response in MCBC on the right, also calculated as area under the curve, MCBC or CBG was significantly higher in food supplemented individuals, which indicates that food individuals were likely in better condition and able to buffer against their heightened response. So if we look at both free cortisol and CBG or MCBC, across cycle phases, the expectation was that from the increase to the decline phase, free cortisol would increase and MCBC would decrease. But I didn't really see that, and the results seem to change in both directions and certainly do not track the changes in predation risk. Now, if we look at glucose, this is shown a little differently, controls on the left and supplemented on the right. Basically the glucose levels in milligrams per deciliter at each blood sample collected for control and supplemented in each of the 4 cycle years. Food supplemented individuals had lower glucose production in response to the hormone challenge, except during the first year of decline, where they had a higher response. So the response was not consistent, and there was no variation between cycle phases in either treatment group, which again did not really match our predictions. So based on these sort of results, what can we really conclude? Well, basically we found that food supplementation increased hare stress response, but also increased their ability to buffer against it to avoid any deleterious effects. And secondly, we found that indicators of chronic stress did not track changes in predation risk, either measured as predator abundance or predator-prey ratio. And this is really what's most striking. Now, to sort of conclude overall what all this work has shown us and what we've learned about the cycle, my goal was to examine the role of food in the snowshoe hare cycle and identify through which mechanisms it could be impacting demography. I tried to choose an all-encompassing approach by comparing demographic parameters between these control and supplemented individuals across the 3 cycle phases, and really aimed to identify the cause of these differences by measuring potential mechanisms, such as changes in behavior, condition, and physiology. And really what we found is that hares are food limited. Food limitation may lead to decreased body condition, which may cause decreased reproduction. Also found that food limitation may lead to higher foraging rates and selection of open habitat, which may increase vulnerability to predation and reduce survival. Food effects are certainly phase specific, with a stronger response in the increase versus the decline phase. And really most notably from the stress work, stress effects may not be necessary for cyclic dynamics to occur, since I didn't really find them to be tracking our predicted response, but yet the cycle still occurred. So we can say that food and predation, both direct and indirect, are likely interacting to cause the cycle. Specifically, food likely plays an important role in halting growth and setting peak densities, and a smaller role during the decline phase when predation is the major factor, and food may still have a small impact on reproduction at this time. So these results really provide evidence for food as an important component of the hare cycle, as well as evidence for the manner in which food is impacting these demographic changes. And more broadly, to sort of step back from hares specifically, food supplementation experiments, I think this research really makes a case for trying to move towards individual-based food ads when trying to look at demographic changes. We were able to feed individuals without the unwanted biases that may occur with large-scale manipulations. So researchers should maybe consider using this method in the future. And I think there's a lot to be learned as well from revisiting some of these classic large-scale studies with this new method, as it's likely to give us new insights as we continue to gain access to more advanced technologies. And with that, I would really like to thank all of the collaborators and contributors. Would like to thank the Champagne-Atish-Kluane First Nations, whose land this work takes occurs on, as well collaborators. I won't go through the list, but Stan, Rudy, my co-supervisors, as well as everyone who works in Kluane, and the funding sources definitely have made this work possible, as well as many people at the University of Alberta and at Squirrel Camp for advice and support along the way. So with that, thank you very much. And I'm here as a visiting postdoc, so I really hope I get a chance to meet some of you in person, or at least chat further. So anyways, thank you very much.
Cool, great talk, Yasmine. Yeah, I really enjoyed that. If people have questions, if they could just throw them in the chat box, just say you have a comment or question, and yeah, we can go in order of appearance on that. And I guess while people are thinking, I guess I could ask a question first. Yeah, really glad you had some endocrinology stuff in there. That's really cool. So my question just from that is, do you think there could just be an overall lack of chronic stress response, and maybe the hares that are supplemented with food are just changing their HPA axis set point to, I don't know, to adjust for that higher quality food? Maybe like their higher cortisol levels are associated with more fat breakdown or glucose formation just because of the food?
Yeah, so that's something I tried to, dive into the literature to figure out why with the supplemental food would they suddenly have such higher stress. And really, it seemed that although they had the higher stress, I think it's important, or I should say higher cortisol levels, I think it is important to remember that they also buffered against it, which is really interesting and kind of showed that they were in better condition. And kind of reading through the literature, there seems to be other instances in other species where individuals in better condition did have higher cortisol levels. And so it might not even be necessarily that it's a negative. And I know there's a bit of a shift in trying to move away from just considering cortisol as, if it's high, it means you're in a rough shape. There's actually, sort of many more multifaceted ways we can look at it. And so this is kind of a first step reevaluating these things, but I think really we should move to measuring more all-encompassing, because, if you only measured cortisol, you might walk away thinking, oh, they're actually in worse shape, but then you look at CBG and all of a sudden, oh, wow, they're actually in better shape and maybe better prepared. So it might also be a bit of a shift in how we look at these things, but you're right, it could be diet influence, but really it's hard to say. It was a bit of a surprising result, I would say. Yeah.
No, that's cool. And I'm glad you're accounting for just higher cortisol and not being associated with stress, because that gets a little annoying after a while. Just being like, oh, the cortisol levels are so high, they must be super stressed out. Not all the time. So, yeah. Yeah,
and it's interesting to think about too, because it didn't really track predation risk or anything. So especially in the hares, there may be other factors that are more influential to what sets their stress levels than just food and predation. So that's still something to be explored, because we've kind of gotten stuck on just solely predation risk. So yeah.
Cool. Awesome. Okay. Looks like Levi has one.
Okay, great. Yeah, thanks for that talk. That was a great, very interesting. I love thinking about glucocorticoids. I'm going to try to phrase this properly. Okay, so you found that the food supplemented hares then had higher glucocorticoid levels than the unsupplemented, and that doesn't really track predation risk. But I guess there's some thought that glucocorticoids can be associated with reproductive effort, or lactation and other things like that. And if they are, I don't know if it's enough of an increase in litter mass or mass of the individual offspring that it would cause an increased investment in reproduction to increase glucocorticoid levels. But is that something like that possible? Or?
So I guess I'll just go back a little, because this is maybe worth sort of remembering that A lot of the measurements I did happen in the winter. So all of the hormone challenges too happened in February, but hares don't actually start reproducing until May. Well, technically they become sort of scrotal and start reproducing in end of April, but they don't give birth till mid-May. So it's unlikely that sort of reproductive investment in the summer would, backtrack to influence what was going on in February. If anything, February results would likely more sort of potentially predict what's going on in the summertime. It's interesting because so we did find in doing sort of subsequent analysis that I didn't present here that body mass was actually a really good predictor of what your reproductive output was going to be. But when we look at body mass and cortisol, spoiler alert, we haven't really put this out in the universe yet, but body mass and body mass changes and cortisol did not correlate at all either. So it really seems like the story with hares can really be told without invoking a chronic stress thing, which sort of really goes against what my entire research group has really been doing up until I came here and made things difficult. But yeah, it seems like it's really based on behavioral changes that are affecting foraging, affecting, body condition, which then affects reproduction and maybe survival. So yeah, it seems this chronic stress component maybe sometimes plays a role, but seems like it's not necessary in order to make the cycle happen is sort of, I guess, what we can conclude. But yeah, because previous studies found really strong results. So it just seems like maybe it's not always present.
Thanks. Interesting to think about.
Yeah.
Cool. Sorry, Jack, I skipped over you if you want to go for your question.
No, keep skipping me. I've talked with Yas plenty. I can save mine till the end if there's time.
Cool. We'll move on to Emily then.
Okay, hi. First of all, great talk. I think that was a really cool way to look at how food limitation will affect a population in the winter. I guess one question I have, it's probably a step past the kind of research you presented, but what about, I guess, the next step I would think is how much food is actually necessary to see those changes? Where, I know you had videos and you're able to track browsing. Is there any way that you're able to kind of take that and get an estimate of how much food is being consumed?
So that's a really good question. And that's totally what if I were to keep doing this work, that is 100% what I would move towards. Because really, and this might sound maybe a bit funny to people not in the snowshoe hare world, but really everyone had moved away from food. And even when I would present my results to sort of the snowshoe hare world, people still didn't really believe but food doesn't matter. So even my experiment sort of seems simplistic and a sort of all or nothing, but this really was to try to redo previous experiments and see okay, if we give them the best of the best, can we change demography at all? Or is it really, not a thing? And so that's kind of where my experiment was as a first step. But I totally think that the next would be sort of having multiple treatments with varying amounts of food to see where that threshold might be. And really the next even better would be moving on to quantifying the landscape more and understanding more food availability. And I know that there's actually quite a few people working on these types of questions already at MUN, but yeah, I think that's really sort of the next steps is getting more fine scale and more trying to understand what's naturally out there and naturally occurring. So yeah, no, definitely the next step.
Well, thank you. It was a really awesome talk. I appreciate it.
Thank you.
Cool. And we have one from, I believe is Evan. If you want to, I don't know if you can turn your mic on, Evan. Let's talk to that.
Yeah, I'm here.
Perfect.
That was a fantastic talk. It was interesting and well, lots in there. So one of the things that you showed was the birth weight of the young.
Mm-hmm.
And I wanted to follow up on that and see how much does that matter to the demography, and what are juvenile survivorship rates like, and do they vary with phase?
Yeah, so that's a great question. And really that is an area where I wished I could have devoted more time. It would have involved me being in the field year round. So that is one sort of gap that I wish I had, sort of following babies after they were born to see how they survived and potentially recruited. But what we do know, if I grab sort of at pieces that other people have worked on, we do know that the bigger you are going into the fall, the more likely you are to survive the summer. And if you're born bigger, you also have a better chance of putting on weight each week. And so if we think of the sort of bigger scale, there is reason to believe that these babies born in these first or second litters, if they are able to be bigger faster, they can put on more weight, be bigger in the fall, and then recruit better to reproduce the following spring. So I don't have a great answer, but that's sort of, we have some indicators that being bigger is actually better.
Great, thank you.
Jack, looks like you have time for your question. If you want to go for it.
Cool. Yes, excellent talk, Yasmine.
Thank you.
I was wondering, so like my impression of the hare lynx cycle is like kind of the magnitude and intensity varies with latitude, like it's less dramatic in further southern populations.
Mm-hmm.
And I was wondering if you could philosophize if like this food habitat usage behavior stuff, like if that would vary across the landscape and maybe like explain why the magnitude differs.
That's a good question. So there is potential because obviously as we move across the boreal forest, hares are eating different things in these different sort of smaller microhabitats. Even between, you know, the Yukon and here in Newfoundland, they are eating different things. So it's hard to sort of generalize, I guess, across the cycle, but there is still the potential that as numbers get high, especially in these sort of less productive northern environments where there's maybe not a lot of high quality food, you're probably seeing similar patterns of high density resulting in not enough food, resulting in predators taking over and so on. But it does get complicated as you go to the south because the more predators you throw into the system, that's sort of always the argument for those more southern latitude hare populations, is that when you have a mix of many generalist predators, you lose the cycle because hares never really have a chance to jump up in density. There's always somebody there to sort of eat them and bring them back down. I don't know if that answers your question, sorry.
Cool. Yeah, it looks like Adam has a question. Want to go for it, Adam?
Yeah, thanks. I absolutely loved your method of getting individual food instead of population food. And so often in field ecology, it's like some clever modification of like a commercial product. And so I just loved it. Was there any lag in like learning time? Did the hares get it pretty much immediately, or did you have to wait a little while for them to just learn how to use the thing?
Oh, that's a good question. So actually these feeders are really incredible. We even like tested them in freezers at the university just to make sure they'd stand up to winter temperatures because it said right on the box, do not use outdoors. And they totally worked, so that was amazing. But yeah, they actually came with like a learning function. So you could program it to, let's say, only open halfway and then close again to kind of teach the hares a little bit to get comfortable. But I was really surprised to find that the hares within, let's say I gave them about a week, but within 3 or 4 days, they were already going to the feeder and pretty comfortable and not shy about the door moving or anything. So they actually learned really quickly.
That's awesome. And like a perfect solution too. That's so amazing. Anyway, thanks very much. Great talk.
Thank you.
Cool. Any other questions from the audience? And feel free to just unmute yourself if you have a burning question you want to ask Yasmine.
And if anyone ever does want to chat later, like please contact me. I don't know if maybe there's ways you could find my contact info, but yeah, I would love to talk further with everyone in the department. It's too bad we can't all be together. So I'd love to, if people have more questions, contact me direct if you don't want to ask now.
Cool. Well, yeah, looks like we can end it there. And I just want to thank you again. You were very last minute, and we appreciate you filling in. So yeah, hopefully we're excited to see what you do here in Newfoundland with the bun work. So yeah.
Well, thank you very much. Yeah, thanks for having me. This was fun.
Right on. Alrighty. Yeah, everyone have a good Halloween tomorrow, and we'll see you next week.